Thank you to the organizers for putting this together. It's a very exciting opportunity to give you an update on the work we've been doing on Alzheimer's disease. As we've been talking about with looking at the trajectory of the disease, we know this has a very long latency period. There's changes in A beta and tau that happen early on. And the idea is that these things are happening in a systemic manner. Our brains, having these plaques and tangles deposit. And the thinking is that the A beta process starts earlier and followed by P tau. But if you're looking at adult populations, people in their 30s or 40s, the P tau becomes a very good marker of what's starting to happen in the disease. And I think this is very important as you're going through and tracking what's happening. So about 10 years ago, when I was at Emory University, I was collaborating with Dean Jones, and he was using these high-resolution methods, and we did this initial study in Alzheimer's disease. And this was a relatively small study, less than 150 people, MCI, AD, and it was this first study that convinced me that this would work. So in this initial analysis, we saw changes in dietary components. These are like pepper derivatives. We saw things like glutamine changes, what people had reported before. We saw this metabolite of DDE, and this was very exciting because Jason Richardson had published a paper a couple years earlier where he did a pesticide analysis in AD and had shown DDT metabolites were elevated. So I had this sort of external confirmation that our untargeted approaches were picking up many of the main components. And then also it was interesting that we detected some of the drug metabolites that are used for the disease. And actually didn't include it in the data as kind of thinking it was extraneous. And in retrospect, we realized that was kind of a mistake. And we've now shown through other projects is we can now measure nearly 1,000 medications and over 10,000 of their metabolites. So we can do systems pharmacology using these high-resolution mass spectrometry techniques. I think essentially establishing a field of pharmacoexposomics that can balance pharmacogenomics. So don't have time to talk about today, but it works. So we've been talking about this, about how many people we need. I think for a given disease or condition, we do not need 1 million people. That if you have deep phenotyping and multi-omic data on hundreds to thousands of subjects, you can get great biological insight. And the high-resolution mass spec, because it's so good at picking up that small molecule space, whether it's exogenous or endogenous molecules, you can capture a lot of information. So in 2018, I moved to New York City. One of the first people I interacted with was Richard Mayu, who's the chair of neurology, who had been working on several large AD projects for many years. And so he said, well, we have this project YCAP in Washington Heights. It's a longitudinal aging study. And we decided to do a pilot with this with controls and disease. It was a third African-American, a third Caribbean Hispanic, a third white. And we were able to easily separate cases from controls, not really a surprise, but could also separate people by their racial ethnic background. And Richard was surprised by that and really questioned. He's like, we've never seen any difference. The genetics aren't different. There's nothing else there that we've seen, but they know that they have differential vulnerability to Alzheimer's. And I said, well, it could be something as simple as diet, because we pick up all sorts of dietary constituents. And he's like, well, that's not possible because we did a dietary inventory. And I was like, that's not biochemistry. Like, that's what we're doing. We're measuring all these molecules. And so we were able to show different pathways were being disrupted in these populations. Again, this pilot study was 160 people, but he had 3,000 people in the cohort. So we wrote grants, we got them, and started doing more. And so one of the things we had shown from this is that besides having changes in and measuring a lot of these environmental molecules, we also had residential history on all these people. And so we could map their residential location to things like air pollution. And so we had information on all sorts of air pollutants, including PM2.5, and look at the sort of exposome-wide association study in correlation to those external molecules. And just one of the quick findings from that was that we found glutathione intermediates elevated in the people that had higher exposures to PM2.5. Like, so you're connecting these external exposures to what's happening in the biochemistry in the person, in the individuals. So I think this is very important for exposomics, is it's not just saying we can measure things inside people, we can couple them to what's happening outside. And with this geocoded information, I was being patient to get up to this more 3,000 population, is there's all sorts of external factors we have data on. We have things on weather patterns and radiation and all these different factors here that we're now going through this analysis and bringing in the geospatial with the high resolution mass spectrometry. We've been really doing a lot of deep characterization of these cohorts. I don't have time to really go into all of these different papers, but for example, we've been doing a lot of MET-QTL analysis in this. And what we said is like, why don't we do an EQTL? Why can't we do like the environmental factors and how it's matching up with that? And this has been working beautifully. Again, don't have time to go into it, but it shows that we can use and leverage many of these methods that have been used in metabolomics to pull it in on the exposomic side. Another area that I think is something I haven't really heard much about at this meeting is the use of some of these predictive models like AlphaFold. So this was a project we were involved in where we were looking at Alzheimer's disease, looking at the QTLs and Mendelian randomization to identify the potential structural variants that would come from those modifications, and then using AlphaFold to predict those changes and the functional outcomes of it. And this also worked very nicely, and my thinking was that you could easily do these things to look at the exposome target interactions. And it's something that toxicologists are kind of starting to look at some of the AlphaFold approaches here, but I think it's something that in exposomics, we really have to take advantage of those technologies to do more of the computational connection to it. So the goal of this was really to get these sort of exposome-wide association study approaches in this mass spec small molecule space. And this is what the data look like when you come down to it, is we can look at all the molecules that associate with the clinical diagnosis, like with AD. And when we started these projects, these new p-tau biomarkers weren't available, but we've gone back and done that on the entire cohort. And what we found was that if you look at people that had clinical diagnosis and the p-tau cutoffs, it was a different group. And this was very interesting because Richard Mayu, who'd been studying Alzheimer's forever, he's like, we've been studying people all this time. He's like, we're misclassifying like 10, 15, 20% of the people that we're saying have Alzheimer's, they have dementia, but they don't have the pathological things you think you'd be seeing. And so when you start reclassifying the people by people that have clinical dementia and the biomarkers, the data totally change. And if you look at what happens, like all these blue dots you see associated with p-tau 181 are small molecules that are somehow related to p-tau 181. Are they drivers of it? Are they downstream actors? Not sure, but they're all things we can look at. These are all, we don't know exactly what all of them are, but we know how to find them and figure out what they are. And we can do the sort of network analysis, and you can see here that even though the p-tau 217 is being used clinically, the 181 appears to have the most biological significance. It's the one that, as it changes, more is changing in the overall biochemistry of the individual. We also have, one of the biggest things we saw from this were these lyso-PCs that were elevated. And again, it wasn't like we found one single biomarker. We were finding the entire family of the lyso-PCs changing and seeing this across time. This is also a longitudinal study. We have samples every 5 years from them over 3 consecutive in this population. But we saw this lyso-PC population first in a FIGA, which is, this is a cohort that Richard developed after seeing this effect in the Caribbean Hispanics in New York. He went to the Dominican Republic and started a new cohort in the DR to look and see if you could separate that sort of genetic background and where people were living. And so we'd seen this population in this lyso-PC effect, but we wanted to see it in other populations. And so we went to the YCAP study, which we've been doing, and saw many of these changes also in those lyso-PCs, and then went to ROSMAP, which isn't even our population, it's just collaborators, and they had brain pathology with these as well, and it's a white population. And we see all of these patterns in these lyso-PCs changing in this third cohort. So again, this idea of replicating across the cohorts and seeing these pathway changes, it wasn't always the single molecule that was the same throughout. It was the patterns of those classes of molecules that were changing. And what's important is that these lyso-PCs, besides being associated with that p-tau and the disease progression, they play a key role in transporting the protective polyunsaturated fatty acids into the brain. So from a mechanistic standpoint, this is like where these modifications that we're picking up are helping you figure out like how these things are being transferred into the brain, and then actually seeing those things in the brain that we saw in the ROS map. And this was an ApoE dependent effect in that, which was not surprising. It was actually a good confirmation to see. And so also we're going through and looking at these very detailed identifications, so you get these long tables with all these mass to charge ratios and retention times. And so we're going back and looking through all this data. And one thing we saw in this was a lot of changes in creatinine. And so we started thinking why does creatinine keep showing up in a change from this? It's so you have a problem with renal function or something, and we know people that have kidney disease often have worse outcomes of many different other conditions. But it got us to start thinking about how we look at those alterations in creatinine. So Vrinda Kali on my team went through and just did this analysis focusing on the creatinine. And what we found was there was this very curious separation. There's a subpopulation, it's almost half the population, whose creatinine status stays steady throughout the time 0, 5, and 10 years. And then there's another group where it's starting to elevate. And so you have these creatinine levels going up over time. We did try to go back and see if they had clinical chemistry creatinine, which they don't, but these are level 1 annotations. But again, we're picking things up that aren't in the medical record. There's a little bit of GFR sorts of data, but it didn't seem to link with that. But as you see from this is we're looking, because we've been following these people up, we're not talking about 1 or 2 years. I mean, we have follow-up of these people from 15 to 20 years. And so you're getting this longitudinal information and seeing that the people that are in this higher group don't seem to be doing as well. So you have a population who you can pick up from a blood test that are going to have a faster rate of progression of the disease. So it's an interesting clinical observation, but mechanistically, how is that happening? And so as we looked into this more, we see here that, again, you saw this shift, is that people were left shifted if they were in this creatinine group. And then, so the question is what could this be? We know that uremic toxins can have a lot of adverse effects for people with kidney disease. They can do things like disrupt the blood-brain barrier. There's many other cardiovascular increasing risk of stroke, but they also lead to the increase in uremic toxins. And for example, the indoxyl sulfate, ADMA, and chynuric acid, we saw all of those elevated in the population with this lower kidney function. And so I was just looking at this last week, and it was interesting because Brad had been on this paper. But if you think something like uric acid is bad, is that if you went through a trial of a drug that reduced the production of uric acid, like allopurinol used for gout, you might expect an effect on AD. And sure enough, the allopurinol trials tend to show that it decreases the incidence of AD. And so the question is there a mechanistic connection here from these uremic toxins to what we were seeing? Now, the data I was just showing you, I got a week ago. So we haven't had a chance to follow up on these individual molecules, but it gives us some very clear clues that are linking up to the disease progression that allow us a way to go through and test many of these hypotheses. So the one thing that I'm showing you a lot of this data on this part is coming from our LC format, but we've also done gas chromatography analysis on all of these same individuals. And we're in the process of doing that analysis. And again, so we have these 3 time points, and so we can look at how very persistent organic pollutants, are they changing over time? Are the more short-acting ones doing that? And just as part of our initial analysis, we went through and did some non-negative matrix factorization, and we are definitely seeing effects here where, again, these alterations with the p-tau 181 are changing. And we know, like on this preliminary analysis, that the furans and fluorinated molecules have been associated, the phthalates and PAHs, and also PCBs and some other pollutants. So again, we're going to figure out what those are. We'll have this data in a matter of weeks to months. But there's molecules there that are highly associated with the p-tau. And then it gets back to, are they drivers of it or are they downstream from it? And those are things we can do. There's other ways of testing this and following up with it. As far as the effect with the creatinine, if you want to see like if there's a causative relationship between renal function and AD, that's a different study. You go and study a population that are being followed for renal disease and look and see what they're doing for AD. And so it gets back to the bringing together multiple cohorts to ask the questions and do the follow-up experiment to look at that next part of it. And so we're not up to 1 million people yet. Like we're able to do this by relatively moderate-sized cohorts. One of the big challenges we've been talking about in the last couple of days is how we deal with these combinations of exposures. Like if you're looking at hundreds of molecules, like how do you deal with that? And so this is from a totally different study, but also done at Columbia, Yakov Stern. He'd been looking at cognitive aging. And so he has this very elegant study where he's been following people. He has people, like 50 people at each age group, age 20, 30, 40, 50, 60, 70, 80, healthy controls. He brings them in every 5 years. He subjects them to a battery of cognitive tests, like pencil and paper, in a functional MRI. So he's getting brain pattern changes as they're doing this, but he took some blood from them, which is like, I always want, like I test it. And so we went through and did a targeted GC mass spec analysis and looked at hundreds of these compounds. And we could show, like by using BKMR, that these clusters of environmental chemicals were associated with these various reference behaviors that occur in the brain function. And so we're getting to this point of like studying these things in combination. And I think these sorts of approaches should work very well on that Alzheimer's side. And the fact it's cognitive decline makes a lot of sense too. But we definitely have seen these things where many of these more legacy compounds appear to be associated. And we've also been doing analysis of the chemical profiles and how they relate to the structural things that are happening in the brain. So again, like we can make these connections back and forth from that by having these very well curated populations. And so again, we're doing more follow-up on this. This paper, I think, just came out a few days ago. But again, like getting back to like testing these hypotheses and looking at causative sort of aspects is, it doesn't always have to be done in a cohort. So my lab had done a lot of mouse work for many years, but several years ago we brought C. elegans, little worms, into the laboratory. And this is an example of what we saw with that elevation of the DDT metabolite in the human population. Again, we'd also seen that from Jason's work. And so at the time, my graduate students said, well, what if we take C. elegans and put in the tau gene, and you could actually put in a version that aggregates and doesn't aggregate. So you have this really nice functional outcome from that. And then treat them with DDT and metabolites of DDT, and then use the high-resolution mass spectrometry, one, to look at the metabolites of DDT, which many of them show up, but then all of the downstream biology that occurs in the worms, because we can use these high-resolution techniques to essentially do like the metabolomics in the worm, and it's the same methods we're using in the humans. And we'd seen many different changes in bioenergetic pathways. Looking down here at the bottom, like we're able to do things like seahorse analysis to look at the mitochondrial function in the C. elegans. But because we're doing this analysis on the same instruments, we can look at what we see in the human CSF, because we have paired CSF and plasma for many of these people. We can look at the changes we see in human plasma. We can look at the changes we see in the worms, and there's actually overlap. And so the idea is we saw many of these sort of cysteine molecules, which we know are also involved in the sort of glutathione redox pathways that are conserved between worms and humans when you're thinking about these tau environment interactions with these legacy pesticides. And so this is like one simple example. I think it does show that, again, like when we think about caloric restriction we were talking about and apoptosis, like these were discovered in C. elegans. Like we do have a lot of conservation of biology, and I think using C. elegans and organoid systems are going to be a very good way for us to follow up these findings we get from these larger cohorts. So I said we don't need 1 million people, but I want 1 million people. and I think it's not so much to say like, oh, like we can't do this. These things are happening. Like you have the All of Us program, which is targeting 1 million. You have UK Biobank that has 500,000. You have Kadoorie in China that's pushing the same thing. And you have the 1 Million European Genome Project going on. And we had many of these people together in Brno, Czech Republic last fall, like talking to the people running these cohorts about how to get exposomics into them. So like we don't need new ones, we just have to get them to include exposomics in what they're doing. And you think to the Human Genome Project, how Craig Venter scaled these things like by having 300 sequencers. Like, give me 50 mass specs, and we could easily get to the point of running 4 to 5 million samples over the course of a 4 or 5 year grant. Like it's not impossible to do. It's just a scaling issue and an engineering problem. And Remy was talking about 1 billion. I could even say $2 billion. Again, I look at people from NIH, which has a budget of almost 50 billion a year. It's not crazy to think about doing this scale of study because you already have All of Us, you have ECHO, you have all these different cohorts that have been funded by NIH. You have the same things going on in Europe. It's just a matter of systematically applying exposomics in a manner that really advances these across all these different diseases. And for example, hopefully you saw Shenchung's poster here from, it's been working with Chirag Patel and Pong Gao. We went through it and looked at UK Biobank, so looking at almost 500,000 people, and did an exposome-wide association study for Alzheimer's disease. And so this is still under review, but like it works. You can look at these patterns in these large-scale biobanks. It's different than the cohorts where you have more phenotypic characterization, but you need to go across all these different modalities. And if you do that, you can get this convergence of the mechanisms that really help you understand what's going on with the disease. So I think that these things are eminently feasible, and that it's not about should we be doing them. It's like we are doing them. We just need to make sure we're doing them in the most effective way that has the best repeatability and impact. And with that, I just want to thank the people in our group that have helped do all these things. I do want to point out that we had this Banbury conference talking about exposomics, and I noted that of like the 25 authors, 7 of them are at this meeting. And this is where we went through and tried to go think about real deeply about how we advance the field of exposomics using an operational definition. And it's something that we've been, I think it did coalesce around many ideas here, and I would encourage people to read that. And with that, I'd be happy to take any questions.
Gary, always really exciting to see the data. So you at the end of the talk, you were talking about how your Venter came in, just bought 300 sequencing machines and really ramped up. But at that point, it was very clear that he could generate nucleotide sequence and that would help to achieve that endpoint. So you're providing us with some very compelling data suggesting that looking at metabolic profiles can reflect the complex environmental exposures that people are exposed to. The question is it ready now to just buy 300 mass specs? Is the preliminary data and the connections between doing these metabolites and different profiles, is it ready? Or is there more preliminary data that needs to be generated in, say, a pilot study? And once the pilot study, you have just convincing absolutely this metabolite or these profile of metabolites give you a signature of exposure to a complex environmental exposure. Is it ready for that? Can we convince funders to come up with the kind of money that it's going to take to get it done?
So I think you can always improve the validation and specificity of the work. We are doing some of these cross-platform analyses right now as part of another project that'll give us a lot of this repeatability aspects to it. But I think this is a place where, as I've written before, is that perfection is not required for exposomics, is that you have all this untargeted data and it's all there. And we might say, yeah, we see this clear signal from air pollution and PM 2.5, but it's just going to keep getting better and better because the data are still there. The raw data are always reminable. But we can show now that, yeah, like for example, we can look at neighborhood deprivation index and see all sorts of small molecules in these signatures that correspond to that. So we are detecting the downstream impact of social factors and physical factors. And so I think it's not like we're just measuring the chemicals, because we're measuring our endogenous biochemistry. And I think the key part is, I think we do have enough to say we can definitely do this layer of it, but we have to be willing to say we know we can go back as we learn more and more and get more in the databases. So I think it is, there's no doubt if you're going to make a large investment, there's a lot of validation and kind of preliminary analysis you want to have, but to do that, you need the instruments in place to do it and to do that sort of rigorous control with that. But I think we're ready to start that.
Great talk, Gary. 2 questions. The first one, I'm intrigued by creatinine. As you saw yesterday, we find that chronic kidney disease itself, due to small vessel disease, because the signature that predicted it was derived from brain small vessel disease predicting kidney disease. So how do you tease apart basically biomarkers, which may be reflecting parallel pathologies in the brain compartment and other compartments versus actually drivers? I like your C. elegans example. I guess the next step would actually be administering creatinine to cells or C. elegans. But in this instance, did you look at CSF, for instance, to see whether levels were elevated in the brain compartment?
So we did look at across that platform, but I think we did do mediation analysis, and creatinine does not appear to be on the causal pathway. That's like where I think it's more probably reflexive of these uremic toxins, or like you said, the vessel damage and things. But these are places where in animal models or like in organoid systems that have that vasculature, we could go through and test those molecules and see that comparison. But it's a very good point, is that you have biomarkers and then you have what's happening there, and we have to be able to do that. The teasing it apart is kind of hard because, like I said, it's relatively new data for us, and I've been talking to a lot of nephrologists, like saying, hey, help me out here. But I think what you point to, though, is that we can't study these diseases in isolation. There's so much multimorbidity. And so even if it is something where people that have chronic kidney disease have a higher risk, it's still good to know why that is. Is it the vessel damage? Is it the uremic toxins? But it's not like we have to totally separate them, because so many people do have both conditions and some other cardiovascular condition on top of it. So I do think we have to get more to this multimorbidity approach, because, and again, like we look at NIH, like we separate by all the organ systems, and it's like we have to study it in more of a systems approach. And which I like about the Exposomics piece is that this is really systems, pharmacology, biochemistry, and toxicology that crosses all of those.
Great. And my second question is about the numbers. I'm very intrigued about the sort of common shared desire of very high numbers. But from my perspective, we have effect sizes that allow us to do discovery in much smaller numbers. I don't do metabolomics on proteomics. So the question is, what are your effect sizes in the data that you've generated, and how much would you predict that you need in order to really validate or cross-validate in multiple different cohorts? So wouldn't it be better to have samples from, across the globe, or at least across the US to get certainty in humans? And instead of putting that money into 1 million, 100,000, however you wish, put the money in validating it and actually model systems to really understand the associations from a mechanistic perspective, and ultimately to develop clinical tests of the most impactful metabolites.
Yeah, so it's interesting, like, again, if you're thinking about specific diseases, I agree, like that approach for much less, we don't need to have the big numbers. We might want to go to 10,000 or something. But then I go back and look at like the proteomic data in UKB and how great it is to have the data on people that have all sorts of different diseases, so you can look at multimorbidity. And so I think we kind of need to do both, is that my dream project would be, there's about 500,000 samples in all of us right now, is you run all of those and you get proteomics and the exposomics, metabolomics on it. And then you also are doing these cohort studies and doing that kind of cross comparison with that because you have these common platforms. So I think, again, it's kind of a long range vision to it, but ultimately we can't study the diseases in isolation. So we need to have these large-scale biobank cohorts to study all of those disease trajectories.
Just to also get back to the first comment on that, I think the thing we have with metabolomics and exposomics that you maybe don't have so badly with proteomics and genomics is our samples will degrade over time, especially if they're being stored. So if we're collecting a lot of samples and they're just going into storage and we're analyzing them in 10 or 20 years, we're not seeing the same signals. And we've seen this again and again for the environment. A few months later, we can't find the same signals again. So I think I'd like to add to your argument that, yes, we are actually ready to start measuring some of these things now, getting at least something on them while they're fresh. We can always go back and remine the data. We can always improve the methods, but then at least we'll build the picture now and we won't be building the picture on degraded samples.
All right.
And
Although it's interesting is that the data Rule showed yesterday on the EPIC PD, those samples were like 25 years old when we got them in our lab, and it worked well because we were focusing on the pesticides, which are unfortunately persistent.
Gary, Sean Morrison from Mount Sinai.
Yeah.
And I know this is a deliberately provocative question, and I'm sorry, my sleep deprivation watching Canada's World Cup game last night, you're going to get it. But my question is, how much more data do we need? I mean, we have, I would suggest, enough data to say that, current pesticides increase your risk of AD. Air pollution, P25, increase your risks of AD. Do we really need a predictive model and predictive risk scores to identify that subpopulation? Or should we be spending our time, effort, and money on the political side where we start tackling limitations or regulation, which in the US is a bad word, rather than continuing to invest in the science? I guess my question is, how much more data do we really need?
So, I think that we can make some sound policy decisions now for that part of it. But boy, on the scientific side, there was discussion about cigarette smoking yesterday, is that, there was a group that identified a gene that modified that effect, SV2C. And we knocked that out in mice and showed it controlled dopamine release. And now we have a drug discovery program on that to treat Parkinson's disease with a new mechanism of action that came from the exposome sort of approach of the disease. I think there's many more drug targets sitting through here because we tend to use the genetic focus part of it. But I think exposomics can open up the new targets. Like what are those environmental factors doing? Those are the pathways we might need that future intervention on. So I think we definitely need more data for stratification, for clinical trials, for new drug treatments. But as far as a public health prevention standpoint, air pollution, we know we have to control many of these things. So I think we have the data we need for policy, but for mechanistic understanding of biology, there's never enough. I don't see any hands. Bob?
I think my question is actually pretty easy for you. So I'm a big believer that everything has to be mapped geospatially and temporally, and you have, I assume, address and untargeted data on 3,000 people. Have you considered mapping it and starting an archive that others can add to so we start to see potentially where the patterns are of both disease, possibly even p-tau and amyloid beta, and whether or not we're seeing hotspots and what is there in those hotspots?
Yeah, we're doing that, and I think this is one of my views of the All of Us program is we have that residential information. Is that if we can do this analysis on everyone there, they all become sentinels, right? You have the entire United States covered, urban and rural, if we did that. And I agree that these maps could be very helpful in driving policy and informing biology. Okay, all right, thank you. Wait, another one? Yeah.